Hello, welcome to a video about genetics. So we're going to try and explain DNA, mutation, chromosomes, genes, and everything related to that. Quite a lot of detail, possibly a bit more than you need, but hopefully that will help you understand it. DNA, or deoxyribonucleic acid, is a molecule that has a code. That's all you need to know, really. It's a code. Here's a code. You can't tell what this says, but if I told you that the first symbol was a B and the second symbol represented an E and so on, you would be able to break down the code. You'd be able to crack the code and understand what it means. And that's what faced scientists when they were faced with the genetic code. They had a molecule that had regular shapes in it, and they just needed to interpret the shapes. So let's start at the beginning. DNA is the genetic material, and it's found in the nucleus of every cell, with one or two exceptions. In the nucleus, DNA is a genetic material, and it forms chromosomes, but it is essentially a long, single thread of a molecule. Here we see one model of what DNA can look like, and what's important is if you see the structures around the outside of the molecule, which form the double helix, they're indicated a bit more clearly here. They are made of phosphate and sugar molecules. The actual code is the layers of molecules between that here are represented by mostly green and black atoms. It's that ladder of molecules in those spaces in the groove inside the double helix structure that contain the actual sequence of the code. So let's have a look at those coding parts of the molecule separately. Now, I apologise for the colour scheme here. We've gone from green and black atoms to blue and grey atoms, but here we are. These are the bases, they're called bases, that sit in that ladder and are the DNA sequence. And they are molecules that chemists already knew the names of. So when they identified them within DNA, they said, yes, okay, we know these molecules. These are adenine, cytosine, guanine, and thymine. And they gave them letters to simplify things, A, C, G, and T. And they are the only molecules within that central part of the DNA, and they are the four bases from which the code is entirely made. So as I've been explaining that, at the bottom of the screen we have been naming the bases with these single letter codes, and that's what we're talking about. DNA is a molecule with shaped areas that repeat, and we name them, and that was where we get our genetic code from. So that's the key feature. DNA works because it has this code, and the fundamental function of the code is the pairing. So the adenine bases pair up with thymine, and the guanine bases pair up with cytosine. That means if we've got a DNA code like this, we can write the other strand, the bottom strand. We know what it is because we always pair A with T and G with C, and that is how DNA replicates itself. It is two strands, the two strands separate, and then the matching code using the A to T, G to C rule is recreated, and you have replicated your DNA. Right, are you still with me? Excellent. You don't need to remember any of that particularly, it's just there to help you understand. Now here we go then with DNA. DNA is a long molecule, 3 billion bases, 3 billion letters in your DNA code, and that means something like this multiplied 1,000,000 times. This might represent one gene out of the 30,000 genes you've got in your body. Now, the DNA is such a long molecule that we have to fold it up, and it's intricately folded into packets called chromosomes. It's too big to fit into one big packet, so we've split it into 23 packets. You have 23 chromosomes. 23 chromosomes contain all the information needed to make an individual. and that is packaged into a sperm or an egg, which means that when a sperm and egg combine, fertilization, you have 23 packets of information from your father and 23 packets of information from your mother. So we have 23 pairs of chromosomes, and the cell picks which genes from which chromosomes it's actually going to use. Now, we kind of draw chromosomes like this, so either a long band with single chromosome with bands on to represent the different genes, or if we draw chromosomes at the moment where they've divided in cell replication, then we draw them as an X, like the two bands are next to each other. The two chromosomes are split but still joined at the middle. The reason we do that is because that's when we see chromosomes most clearly, when they are in the process of being duplicated. This is what that actually looks like in some cells under a microscope. Got two versions here that are cells that are dividing. The DNA is just blue diffuse material in the other cells that are present in the picture. If we look closer still, we can see a picture like this, and scientists can cut out the individual chromosomes and rearrange them on a piece of paper and make them look something like this if they are going to do something called a karyotype, which is to check that all the chromosomes that should be there are there. This is a karyotype of a human, so there are 23 pairs of chromosomes, and the 23rd pair are the sex chromosomes with X and Y, making this set of chromosomes from a man rather than a woman. Quite a common question is how many chromosomes are there in this cell? Now, they're either going to be talking about a normal body cell, in which case there is a pair of every chromosome present, or they're talking about a sex cell, in which case there's one of every chromosome present. So in a human, the answer would either be 23 pairs, 46 chromosomes in a body cell, or 23 chromosomes in total in a sex cell, a sperm or an egg. Now, as a dog has 39 pairs of chromosomes, there are 78 chromosomes in total in a body cell and 39 chromosomes in total in a sex cell. With a frog, 24 in body cells, 12 in sex cells. They will tell you the number of chromosomes, and you will show that you understand the difference between the number in total in body cells and sex cells. Right then, the next thing. You have one chromosome from your mother and one chromosome from your father for each of the chromosomes you have. Therefore, for each of the genes that you have at least two copies. We simplify it and say, let's just assume there's one copy in the 23 chromosomes. There's one copy of any given gene. Therefore, because you have two sets of chromosomes, you have two copies of every gene. The question is, are they the same or are they different? And how does that affect you? Right, I'm going to explain this using a system that probably won't be mentioned in the exam. It's the blood type system. So you have molecules on the surface of all your blood cells, and there are three types. The type A, type B, and type O. And these come from the same gene, but there's three different versions of the gene, and they're called alleles. So there's allele A, allele B, and allele O. Now, you have two copies of the gene, so you could, for example, have two copies of allele A. If your copies are the same, you are said to be homozygous. That means you've got the same pair. Homozygous, you could be AA, BB, or OO. If you have two different alleles, like A and B, you are called heterozygous. Your genotype is heterozygous. AB, AO, BO, for example. So let's consider this situation. We've got Fred, who has 2 copies of the blood group A allele. He is homozygous for A. He has the genotype AA, and the phenotype, that's what he actually shows, is to have blood group A. His wife Julie has the B allele and the O allele. Therefore, her genotype is BO. She is heterozygous, and her phenotype is B. Her phenotype is B because O is recessive and B is dominant. And a recessive gene doesn't show unless it's the only gene there. A dominant gene always shows itself. So to look at all the possible combinations of alleles in any children that Fred and Julie had, we draw something called a Punnett square that looks like this. So we've got Fred's possible gametes, or sex cells, in this case sperm. What alleles can he have in the sperm regarding this gene? Well, of course, he's only got the A allele, so that's the only one that he can have. Julie's gametes, her sex cells, her eggs, can have one gene, and it can either be the B allele or the O allele. So if we fill in the boxes, we get the answer to look like this. And as you can see, there are 2 possibilities. There is a 50-50 chance of the child being either genotype AB or genotype AO, and that would mean phenotype blood group AB or phenotype blood group A. Similar Punnett squares to this can be used for lots and lots of different inheritance questions. It could be inheritance of a dominant disease or a recessive genetic disease or eye colour or earlobe shape or anything like that. So be prepared for those. I'm going to leave you with a slide reminding you about the keywords that you need to use. Particularly difficult for some people seems to be homozygous, means you've got a pair of alleles that are the same, and heterozygous, you have a pair of alleles that are different. You have to have a pair that are either the same or different because you have to have 2 copies of every gene, because you inherit one set of chromosomes from your father and one from your mother.